Hey guys, in this lecture we're going to be talking about two things here. First, we're going to talk about diversity, the diversity in a community. We can also call it species diversity. Really what we're trying to say here is we're looking at the variety of different species or different kinds of organisms in a community or in a biome. And then we'll also talk about this thing called trophic structure, describing in some ways the feeding relationships in a community. We've learned previously about the different kinds of species interactions. One of the more important ones, of course, is predation and herbivory, where one species eats another species. And so if we understand how and which organisms are eating which other organisms, we can understand to a greater extent the role of that organism within the community. And really, we now know that to be the definition of an ecological niche. So we can understand a species niche if we understand where it lies in a trophic structure of a community and how diverse that community is. To first start off talking about species diversity or community diversity, diversity is going to be described or differentiated based on two criteria. The first one will be considered species richness, and we'll define that by the number of different species in the community. So a community might have 50 different species living there. It might have 500 different species. It might simply have 4 different species. They're all communities because they are all areas with different populations of different species, but the species richness might differ from one community to the next. The other criteria that describes species diversity is the relative abundance of those species. You might have a lot of species, but some species might be particularly abundant in the community, while other species might be very scarce on the other hand. So relative abundance is defined as the proportion of each species, or the proportion that each species represents of all of the individuals in the community. So you might have 100 individuals. 99 of them might be of one species. It might not, and then the other would have a relatively low relative abundance. So we can talk about species diversity using two examples. These two examples present us an interesting dilemma. The question that we're trying to answer is which community is more diverse? We're looking at two communities of forests, two forest communities, and really we're only going to look at the trees in those communities, but while we also recognize that we're ignoring all of the other kinds of animal life and so forth. So we're really only looking at the trees of these two forests. We have community 1 and community 2. In community 1, you see that we have 4 species of trees. We have species A, B, C, and D. And we see those species here, A, B, C, and D. All right, 4 different species of trees, and we can see in community 1, each has a relative abundance of 25%. Okay, let's take a look at community 2. In community 2, it's with the naked eye, you can see that species A is much more abundant than the other 3 species. You'll notice that species A has a relative abundance of 80%. 80% of all of the species, all of the individuals in this community are members of species A. Species B and C both have a relative abundance of 5% of the total community, and species D has a relative abundance of 10% in the community. So while both communities have the same number of species present, community A has different relative abundances of those species than community 2. The question then is, which community is more diverse? Well, there's a particular measurement or a particular kind of calculation that we can perform that will help us understand this. The kind of calculation that will be called the Shannon Diversity Index. Let's take a look. The Shannon Diversity Index is simply just a tool that we can use to measure a community's diversity. You'll see that it is simply an equation that creates some kind of number. The bigger the number, the more diverse this community is, and the lower the number, the less diverse the community is. And we calculate the Shannon Diversity Index, and we'll represent that index with an H. So the Shannon Diversity Index, H, is equal to negative, okay, take a look there, and then you take all of your different species A, B, C, and D, and so forth, and you take the relative abundance of each species in a percentage or in some kind of decimal form, so we're going to call that P, multiplied by the natural log. All right, so that's what we see here, ln, the natural logarithm multiplied, of that relative abundance. And we do that for species A. We do the same thing for species B and C and so forth. And we sum all of those quantities together to get the total diversity index, or the total Shannon diversity index. So using this index, using this equation, we can then look back at communities 1 and 2 and measure, using the Shannon diversity index, to see which population, which community is more quantitatively diverse. Okay, so we want to measure the Shannon diversity index of community 1. So we know that the Shannon diversity index is equal to, or is recognized as H. We know it's equal to negative times each of our quantities for each species. So we know that species 1, for example, species A, has a relative abundance of 0.25 times the natural log of 0.25 plus species B has a relative abundance of 0.25, pardon my bad handwriting, times the natural log of 0.25 plus the same thing here, 0.25 natural log of 0.25 once more plus 0.25 natural log 0.25. Great. And our brackets. And we know if we were to use our calculator to calculate or quantify the diversity index for community 1, we would come up with a final answer, a final number of, drum roll please, 1.39. Okay, and so that's our diversity index for community 1. Let's try community 2. Here is the calculation for community 2. Our Shannon diversity index is equal to negative 0.8 is our relative abundance for species A in this community times the natural log of 0.8, 0.05 times the natural log of 0.05 for species B. For species C, we've got another 0.05 times the natural log of 0.05. And then finally for species D, we have a 0.1 times the natural log of 0.1. We add these all these quantities up, and we get a total species richness for species 2 of 0.71. All right, looking at my notes. Okay, 0.71 is our species index, or diversity index of community 2. So comparing the two figures, or the two indices for our two communities, we recognize that community 1 has a greater Shannon diversity index, and as a result, we classify this community as being more diverse. Even though it has the same number of species, those species are represented in a more diverse fashion. All right, so you can use this to measure this diversity of any species or any community. So that ends our first half, or the first topic of this lecture, which deals with species diversity. Second topic of this lecture deals with trophic structure. And what we mean by trophic structure are once again, the feeding relationships between organisms. We'll see trophic levels, we'll talk about food chains, and we'll talk about food webs. A lot of this stuff is intuitive, or we may have already studied it at an earlier date. Okay, and so this will be quick. Best representation of trophic structure is a simplified food chain. A food chain just gives you each link in the feeding relationship along the way. Every food chain will begin with the first trophic level, which we'll consider to be producers. All producers are autotrophs. That means they are performing photosynthesis and making their own food. So we're going to call these organisms producers. Another way of thinking of them, usually they are plants or algae, or something like in a aquatic environment, something like phytoplankton. Okay. The next trophic level will be herbivores. We also call herbivores primary consumers. So we have primary producers as the first trophic level, and the primary consumer is the herbivore. That is the first consumer of an actual organism. Those primary consumers or herbivores are also recognized as autotrophs because they cannot perform photosynthesis and make their own food. So any herbivore, like, for example, a mouse or a rat, any kind of maybe a tadpole or even zooplankton in an aquatic environment, or usually a lot of hooved animals that are grazers will be considered, like deer, for example, would be considered herbivores or primary consumers. Next consumer would be a carnivore. So we're going to call this next trophic level the carnivore, the first carnivore level. But we're also considering them the secondary consumers because they are consuming the first consumers. So in this instance, the snake would be a secondary consumer, eating the primary consumer, which eats the primary producer. Okay, this is our third trophic level, but it is our secondary consumer that we're going to call this. We can also label this like tuna, a secondary consumer, and because it eats the primary consumer. And then finally, our top level carnivores of our food chains, which is the second carnivore in the food chain, but we'll also classify these carnivores as tertiary consumers. All right, they are the third level consumer. We have a primary consumer, a secondary consumer, and now a tertiary consumer. So this vulture, for example, I think that's supposed to be a vulture. It might be a hawk. Looks to be the tertiary consumer in this food chain, whereas this heron here might be a tertiary consumer in this aquatic food chain. Notice this food chain doesn't display a tertiary consumer. Not all food chains demonstrate tertiary consumerism, though some do. Okay, but they are equally valid food chains. Now, we have to also recognize that a food chain is a simplified representation of feeding relationships. It doesn't capture the intricacies of a community, specifically the feeding relationships in a community, because we also know that this mouse here, for example, is not the only food source for this snake. This snake eats many more things than the mouse, and likewise, on the other hand, the mouse is also the source of food for a number of organisms, not just the snake. Maybe some hawks eat the mouse itself. Maybe some fox eat that mouse. A number of organisms eat the same types of food items. So a food chain is only a simplified and really an incomplete representation of feeding relationships in a community. We can have a more accurate, a more precise representation of feeding relationships, more precise representation of the trophic relationships when we look at what we'll call food webs. Once again, another representation of food chains. We see our primary producers, primary consumers, secondary consumer, tertiary consumer, and we even here in this food chain we see quaternary consumers. Okay. Again, the grasshopper eats the plant or the producer, so it is a primary consumer. This mouse eats the grasshopper. It is an insectivore, and therefore it is a secondary consumer. The snake eats the mouse, and the hawk eats the snake. And so we have 5 total levels in our food chain here. The same thing can be said with our aquatic food chain. Once again, let me reiterate that food chains are more simplified representations, more incomplete representations of feeding relationships. And we can look at a food web like this marine food web and see a more complete picture of these feeding relationships in a community. Again, let me also highlight the idea that the arrows, all right, are pointing in a direction of energy flow. We'll talk about this more in a couple weeks. So anytime we see an arrow, we're seeing that the energy is moving from this benthic invertebrate to the sea duck, and from the sea duck to a bald eagle, for example. Okay, so here we are with our marine food web, and now we can take a look at a terrestrial food web in a grassland like a savanna, like the savanna of Africa. Again, we see feeding relationships, energy flow moving from a primary consumer to a secondary consumer, and from a primary producer to a primary consumer, and so forth. All right, that concludes our discussion about diversity as well as trophic strategies and trophic structure.